Welcome to another episode from the Veterinary Research Channel. In this episode, we delve into a fascinating pilot study that explores a novel approach to stimulating red blood cell production in healthy cats. The research, published in the Journal of Veterinary Internal Medicine in 2023, investigates the effects of a drug called Molidustat on erythropoiesis, the process by which red blood cells are formed in the body. Erythropoiesis is a fundamental biological process crucial for maintaining adequate oxygen supply to tissues throughout the body. This intricate process is primarily governed by a glycoprotein hormone known as erythropoietin, or EPO. In mammals, the majority of EPO is secreted by specialized cells located in the kidneys, called renal EPO-producing cells. These cells play a vital role in sensing oxygen levels in the blood. When oxygen levels are low, these cells increase the production and release of EPO. Erythropoietin then travels to the bone marrow, where it acts on erythroid progenitor cells, promoting their proliferation and differentiation into mature red blood cells. This increase in red blood cell production helps to restore normal oxygen carrying capacity. Conversely, when oxygen levels are sufficient, EPO production decreases, maintaining a delicate balance. The production of erythropoietin is tightly controlled by proteins known as hypoxia-inducible factors, or HIFs. Under normal oxygen concentrations, HIF is continuously degraded by enzymes called HIF prolyl hydroxylases, or HIF-PH. These HIF-PH enzymes act as oxygen sensors. When oxygen levels drop, the activity of HIF-PH is inhibited, leading to the stabilization of HIF. Stabilized HIF then acts as a transcription factor, binding to specific genes and increasing their expression, most notably the gene encoding for erythropoietin. This mechanism allows mammals to adapt to changes in environmental oxygen concentrations. Given the crucial role of HIF in regulating erythropoietin production, researchers have explored the possibility of manipulating this pathway to treat conditions characterized by low red blood cell counts, such as anemia. Inhibiting HIF-PH enzymes can mimic the effects of hypoxia, even under normal oxygen conditions, leading to increased erythropoietin production and subsequent stimulation of erythropoiesis. Previous studies in various animal models, including rats, dogs, and monkeys, as well as clinical trials in humans with anemia associated with chronic kidney disease, have demonstrated that inhibiting HIF-PH can indeed stimulate erythropoiesis, resulting in increased hemoglobin concentrations comparable to those achieved with the administration of recombinant erythropoietin. Based on these findings, the researchers in this study hypothesized that a novel HIF-PH inhibitor, Molidustat, could also increase erythropoietin production and relevant hematologic parameters in healthy cats under normal oxygen conditions. Their aim was to investigate the effects of repeated daily oral administration of Molidustat sodium on hematologic parameters and erythropoietin concentrations in healthy adult cats. The study was designed as a prospective, randomized, placebo-controlled, open-label laboratory investigation. 17 healthy adult laboratory cats were included in the study. These cats were randomly assigned to one of three groups. The first group, consisting of 6 cats, received a placebo, which was a suspension containing no Molidustat. The second group, also with 6 cats, received Molidustat at a dose of 5 milligrams per kilogram of body weight. The third group, comprising 5 cats, received Molidustat at a higher dose of 10 milligrams per kilogram of body weight. The treatments were administered orally once daily. The researchers closely monitored various red blood cell parameters, including hematocrit, red blood cell count, and hemoglobin concentrations. They also assessed respiratory indices, which provide information about the production of new red blood cells, and measured plasma erythropoietin concentrations. Molidustat treatment was stopped if a cat's hematocrit, the percentage of red blood cells in blood volume, exceeded 60% due to concerns about potential microthromboembolic events as reported in previous studies in dogs. Now, let's move on to the results of this study. The researchers observed significant changes in the red blood cell parameters of the cats treated with Molidustat compared to the cats that received the placebo. A significant increase in mean hematocrit was evident starting on day 14 of the study in both Molidustat-treated groups. In the group receiving 5 milligrams per kilogram of Molidustat, the mean hematocrit reached 54.4% compared to 40.3% in the placebo group. In the group receiving 10 milligrams per kilogram, the mean hematocrit was even higher, reaching 61.2% compared to the same 40.3% in the placebo group. These increases were statistically significant and remained so for the entire treatment period. Due to the predefined stopping criterion, molidustat treatment was halted early for the treatment groups when hematocrit levels exceeded 60%. This occurred on day 21 for the 5 mg per kilogram group and on day 14 for the 10 mg per kilogram group. Following the cessation of treatment, the mean hematocrit in the molidustat-treated cats gradually returned to within the normal reference range for cats, which is between 29% and 45%. By day 56, the mean hematocrit was within this range, and from day 70 onwards, it was numerically comparable to that of the placebo group. The researchers also observed that red blood cell count and hemoglobin concentrations followed a similar pattern to the hematocrit levels, increasing significantly in the molidustat-treated groups compared to the placebo group. Furthermore, the study found that mean erythropoietin concentrations significantly increased after molidustat administration on all assessment days. 6 hours after treatment, the mean erythropoietin concentrations in the molidustat-treated groups were substantially higher than in the placebo group. Specifically, after the administration of 5 mg per kilogram of molidustat, mean EPO concentrations range from 79 to 186 milliunits per milliliter, while after the 10 mg per kilogram dose, the range was 310 to 420 milliunits per milliliter. In contrast, the mean EPO concentrations in the placebo group remained very low, ranging from 1 to 2 milliunits per milliliter. Interestingly, the mean respiratory hemoglobin concentration, a measure reflecting iron availability for red blood cell production, remained within the normal physiological range throughout the study in all groups. Molidustat treatments were generally well tolerated by the healthy cats in the study. While some mild abnormal clinical findings were observed in individual cats during physical examinations, these were considered unlikely to be related to the treatment. Mean body weight and food consumption did not significantly change with molidustat treatment. Fecal consistency remained largely normal, with a transient change observed in only one cat in the high-dose group. A slight decrease in mean white blood cell count and mean absolute neutrophil number, slightly below the reference interval, was observed in the high-dose group on day 14. However, no abnormal findings were detected in serum biochemistry profiles in any of the cats. Treatment-related vomiting occurred in cats across all groups, but the overall vomiting rate was low, below 10% in all groups. The number of cats experiencing at least one vomiting event was higher in the molidustat-treated groups compared to the placebo group. Now, let's discuss the conclusions and clinical significance of these findings. This study in healthy adult cats successfully demonstrated that molidustat, a novel HIF-PH inhibitor, effectively stimulates the production of erythropoietin, leading to a dose-dependent increase in hematologic parameters such as hematocrit, red blood cell counts, hemoglobin concentrations, and respiratory counts following once-daily oral administration. These effects were observed relatively quickly, with significant changes appearing as early as day 14 compared to the placebo group. The rapid and marked increases in hematocrit, exceeding 60% in some cats within 2 to 3 weeks, highlight the potent erythropoietic effects of molidustat in healthy cats. The fact that hematocrit levels returned to the normal range after the drug was discontinued suggests that the effect is reversible. Residual effects on hematocrit were noted for up to 42 days in the high-dose group and 35 days in the low-dose group after the last administration. The mechanism by which molidustat stimulates erythropoiesis is through the inhibition of HIF-PH enzymes. By inhibiting these enzymes, molidustat stabilizes hypoxia-inducible factors, mimicking the conditions of low oxygen and thereby stimulating the endogenous production of erythropoietin by the kidneys. The subsequent increase in erythropoietin then promotes the production of red blood cells in the bone marrow. The observation that respiratory hemoglobin concentration remained within the physiological range despite the increased red blood cell production suggests that molidustat administration in healthy cats for a short period did not induce iron deficiency. However, the researchers noted that the study was conducted in healthy, young cats for a limited duration, and treatment was stopped once a substantial increase in hematocrit was observed. The mild decrease in white blood cell count and neutrophils in the high-dose group could potentially be attributed to a physiological shift in the bone marrow microenvironment. favoring red blood cell production over white blood cell production due to the strong stimulation of erythropoiesis. Another possibility could be a hemodynamic shift of neutrophils to the marginal intravascular pool due to the increased red blood cell volume. The pharmacokinetic analysis indicated that there was little to no accumulation of molidustat or erythropoietin plasma concentrations with daily administration. Previous studies have shown that molidustat is eliminated from feline plasma with a half-life of 4 to 6 hours after oral administration, supporting the once-daily dosing regimen and suggesting a pulsatile effect on erythropoietin production rather than sustained high levels. This pulsatile effect may contribute to a favorable safety profile compared to currently available erythropoietin-stimulating agents. Furthermore, given that renal excretion of molidustat is negligible, it may be a suitable option for cats with impaired kidney function, potentially without the need for dosage adjustments. The researchers acknowledged several limitations of their study, including the limited number of data points for pharmacokinetic and erythropoietin plasma concentrations, the small sample size of cats, and the fact that the study was conducted in cats with healthy kidneys. Additionally, they used erythropoietin test kits designed for humans, which may not perfectly measure feline erythropoietin, although there is a high degree of similarity between the two. Finally, because the cats were group housed, it was not always possible to definitively assign vomiting events to individual cats. Currently, erythropoietin-stimulating agents, such as recombinant human erythropoietin, are used off-label to treat anemia in cats. While these treatments can improve the quality of life for anemic cats, they are associated with several potential complications, including iron deficiency, hypertension, and the development of antibodies leading to pure red cell aplasia, a severe form of anemia. Therefore, a novel and safer approach to managing anemia in cats is highly desirable. This study provides evidence that molidustat can effectively stimulate endogenous erythropoietin production in healthy cats by mimicking hypoxia in the kidneys. The once-daily administration resulted in marked increases in erythropoietin concentration, leading to a significant increase in hematocrit. The researchers concluded that hematocrit is an appropriate and easily monitorable surrogate marker for erythropoietin activity in this context. They suggest that the concept of using a HIF-PH inhibitor like molidustat to induce endogenous erythropoietin production in cats with anemia warrants further investigation. Additional studies are needed to evaluate the effects of molidustat in anemic cats, particularly those with chronic kidney disease, a common cause of anemia in this species. Thank you for listening to the Veterinary Research Channel. A link for the referenced paper is in the video description. Remember to test your knowledge by answering the questions in the description, and as always, keep those minds inspired, hearts light, and tails wagging.